sorry for all of you for delaying and disrupting your schedules, but I think maybe you are more looking forward to my topic after that. So, so I will start. I'm very glad for this chance to talk about the comparative analysis of brain connectivity and gene expression divergence in chimpanzees and humans. I have no disclosures. Okay, let's start. Here is where our story begins. Look at the primate family trees, lots of well-known species. The monkeys, gibbons, apes, and of course, we humans have many similar neural anatomy and cognitive functions to some extent. To investigate how we humans become humans, evolution provides a key perspective for understanding this. And this makes us concentrate on one of our close living primate relatives, that is chimpanzees. We all know the human has a brain 3 to 4 times larger than the chimpanzees and much more than the small monkeys. But is a human brain just a big monkey brain or ape brain? And why did not chimpanzees dominate the planet, but to some extent we humans did? Back to our research, here we focus on the comparison with the humans and chimpanzees, our closest living primate relatives, and we share greater genetic proximity and neural anatomy and cognitive functions with us. They are considered as one of the crucial comparative reference for understanding human resolution. Neuroimaging provides a lot to the comparative neuroscience. MRI allows us to acquire data from multiple modalities and non-invasively. And most importantly, it provides an approach to acquire data from multiple species using the same non-invasive method. Although the neuroimaging has enabled the comparisons across primate species, morphological changes in cortical areas cannot fully explain the evolutionary adaptations between species. And from another perspective, evolutionary changes in the anatomical connections provides another explanation to the functional difference between species. But however, comprehensive whole brain connectional analysis and comparisons between the humans and chimpanzees are still lacking. This partly due to the lack of the comparable brain atlas, cross-species brain atlas. We know the brain atlas provides a map for locating biologically meaningful subregions, and comparable cross-species atlas provides to facilitate this comparison within a common reference system. However, inconsistency in the modalities and scales in constructing previous atlas render these comparisons very challenging. Recently, connective-based parcellation using anatomical connections successfully delineate subregions in humans and monkeys. This demonstrates the feasibility of parcellating the chimpanzee brain using diffusion MRI data, and such an atlasing approach also enables meaningful comparison with these species. So based on the aforementioned question, what we did here could be divided into three main parts. First, we developed the Chimpanzee Brain Atlas, and when compared the connectivity divergence between humans and chimpanzees. Finally, we move on to the investigation of the genetic association analysis with the divergent connectivity between these two species. So here we use 46 chimpanzees in vivo data, MRI data from National Chimpanzee Breed Resource, and following the connectivity-based parcellation framework used previously with humans. We first defined 19 cortical and some subcortical seed regions, and taking an initial region for a seed, we perform tractography for each subject, and resulting in a whole brain connectivity matrix. We calculated the similarity matrix and conducted spatial clustering to define the optimal number of subregions, thereby obtaining a whole brain parcellation. We divided the chimpanzee brain into 100 cortical and 22 subcortical subregions per hemisphere. The atlas is now open through our GitHub repository and also for the visualization. Although the cytoarchitectonic data for the whole chimpanzee brain are not available for alignment, we here use the data from several well-described regions for the validation and found a good consistency between the histology and connectivity-based parcellation. We also show that Chimpanzee Brainnetome Atlas achieved the best performance in the structural connectivity homogeneity. The second part, we move down to the connections in between the chimpanzees and humans. Here we follow the protocols by Catherine and her colleagues to reconstruct the homologous wet matter tracks for each human and chimpanzees, and resulting in a regional connectivity blueprint. The rows in the blueprints indicates region-to-track connectivity patterns for each subregion. Using the connectivity blueprint, we could investigate the connectivity divergence between these two species. For each subregion in humans, we compare its connectivity profiles with all of that in chimpanzee brains and find the region with the minimal KL divergence. The higher value in the maps indicates that the region in humans has a more dissimilar pattern with that in chimpanzees. In another way, we can say that this pattern may maybe not represent in these close phylogenetic relatives. As the figure shows, we found the regions with the most different connectivity patterns were located in the middle and posterior temporal regions. And thanks to the fine-grained atlas, we could distinguish the difference in heterogeneous regions like the Brassenius and Insula, just as Dr. Revan showed before. And most interestingly, we found very highly dissimilarity with the expansion of surface area. This might indicate that the connectional changes reflect the unique aspect of brain reorganization. We're always very interested in the asymmetry of these connectivity patterns. So we here calculated the KL divergence between homotopic subregions of the two hemispheres and for two species. Higher value in the map indicates higher interhemispheric connectivity difference between the hemispheres. We could also examine the track distributions and contributions to the subregions. And since the data from the two species were not in a common space and cannot be compared directly, so here we used an alignment technique based on metadata to transform the data from two species. Here we focus on the human-specific asymmetric patterns by calculating exclusive OR maps. And based on the connectivity divergence map I showed in the last slide, we can find the regions including the posterior temporal lobe, posterior IPL, dorsolateral prefrontal cortex, and posterior orbitofrontal cortex showed marked connectional asymmetries that likely emerged during evolution. The final part, we investigate the association between the connectivity divergence and genetic expression using the ALA data. We here use PLSR to identify the most relevant genes with the divergence map and input them into the enrichment analysis. These genes were enriched in the excitatory neurons, which are involved into the cortical projection circuit construction, and they are related to the neuron projections and synapse formations. These genes also overlapped significantly with the human accelerated genes, and more genes showed evolutionary rate greater than 1 than that in monkeys. Finally, we show that genes express differentially in regions, especially with divergent connectivity patterns. Significant differences were found in the supratemporal cortex and dorsofrontal cortex, but not the V1. To conclude, I want to sum my main points today. We first presented the most refined atlas of the chimpanzee brain to date, the Chimpanzee Brainnetome Atlas. We compare the connectivity divergence and its asymmetry between humans and chimpanzees at a fine-grained level. Finally, we show that associated genes has differential evolutionary rates and expression patterns between species, and these genes are related to the connectivity. Based on the major projects program ongoing in China, we're dedicated to mapping the brain from multiple species, and we want to extend the comparison to more evolutionary distant species and hope for the interesting discoveries. Finally, I want to express my special thanks to my supervisors, Professor Ling Zhongfan and Ceng Chu, and all collaborators and all members from our lab. I have a poster session today and tomorrow afternoon. Please also feel free to contact us if you have any questions and wanting for any discussion. That's all from me. Thank you.
Thank you, beautiful work. The question that we have for you is, do you think that these parcellations will be useful in understanding function?
Yeah, of course, the connectivity-based parcellation is actually could be used to identify the regions and provide a map for locating the functional regions. Unfortunately, in this work in chimpanzees, we didn't have the functional data, but I think we extended to the other species like macaques and monkeys. We also use that to compare with the functional and behaviors. It could have a good consistency.
Lovely. Thank you very much.
Yeah, thank you.